Hello everybody, I'm Nikolai. I'm a scientist, but I'm also CEO of a company, Synvect. We essentially founded this company from University of California, San Diego with my colleagues to bring some of the genetic tools we developed in academic setting and hopefully bring them into the field and start saving lives. And the two tools I will talk about today, it's a separator and NextGenSIT. Mosquito, of course, is the most deadly animal. I don't need to kind of repeat the same message, but maybe the interesting message I want to convey that mosquito numbers and mosquito-borne disease cases are actually increasing, and maybe they're increasing more dramatically outside of Africa. In US, I'm coming from California. We had first cases of dengue ever reported in California history in 2024. There are a lot of approaches to deal with mosquito-borne diseases, but many of them are kind of failing, especially insecticide. There is a rampant emergence of resistance among mosquitoes, but also mosquito nets. I read a lot of papers that Anopheles mosquitoes' behavior of them is changing. They start biting at dawn and dusk, not at night, and then that nets is also not effective. And then there is very expensive methods which were used by US after World War II to suppress mosquitoes, like essentially land control, water drainage. They are very expensive and they are not ecologically friendly. Vaccines and drugs, I mean, they are very promising, but there is always uncertainty about the timeline and efficacy of the vaccine. And then there is like some biological approaches, and probably the gold standard of biological approaches is a sterile insect technique. It's really worked very effectively for fruit flies, screw-worm flies, but it's very hard to adapt it for mosquitoes because it works, but it's very expensive. And there are probably like three major reasons. Because for mosquitoes, you really have to remove female mosquitoes before you make these massive releases. For fruit flies, they actually don't absorb them. For screw-worm flies, they still release them, but they don't absorb them. They don't need to do it. And then there is, because you're releasing adults, and for mosquitoes you have to release adults, it becomes very expensive, you cannot really shoot them, and the efficacy is low because the fitness of mosquitoes is affected by radiation. And I don't need to kind of go over this slide, but it works in many mosquito control districts in the US, actually under pressure for local communities. They adapting these not effective, expensive method of controlling mosquitoes, but they just want to stay away from pesticides. And essentially what Synvect does, Synvect combines the safety of traditional radiation-based SIT with the precision and efficacy of a CRISPR gene knockout. We're not using CRISPR to any gene drive, but we just use CRISPR for essentially cleavage of DNA and mutagenizing genes and knocking them out. And at the core, essentially, kind of these two products are mosquito eggs. And there are two types. One is a separator, which is a sex-sorting genetic strain. And this kind of product by itself, but it's one of a component of a final thing, which we think would be very useful for mosquito control. We call it NextGenSIT. And this is just mosquito eggs, which you can deploy as eggs. And from them, will come out completely sterilized and sex-sorted male mosquitoes. And because we're using eggs, we essentially have now scale of production, because every female in the lab will lay like a bunch of eggs multiple times. And then it becomes also very effective because fitness of these mosquitoes is preserved, and I will explain why. And then it really becomes a product, it's not a service, because if you're doing conventional SIT, it's a service. Somebody has to come and release mosquitoes twice a week, where here you can deploy eggs yourself in the backyard. So first system, separator, sex-sorting genetic cassette. So here you see a genetic map. Essentially it's just two fluorescent proteins. One is red fluorescent protein that's expressed in both sexes, males and females, and it's also expressed from an embryo up to adult through the entire development. And the second color, green GFP, it's expressed only in the males. And it's expressed in the males because we put this intron, which splices correctly, only in the males. And we can see actually in our culture gene andromorphs, because for insects, every cell decides almost independently its own sex, and the system is extremely robust. And this picture shows larvae for Aedes aegypti. This is all of them expressed in red. only males expressing GFP, and then we can also image two colors at the same time with filters which pass two colors. This Anopheles gambia, this is L1 lavis and probably 6 or 8 hours old. And you see these guys are males, but there are females too, and two colors imaged together. And then there is Anopheles SEPARATOR, and here it's one color image separate and the other color separate. But sort of what separator allows to do now, it allows to do very high throughput and high precision sex sorting. And in the context of conventional SNT, a lot of resources can be saved now to produce 2 times more male mosquitoes because females are moved earlier. And then it's a real platform because we built it in Aedes, Anopheles, and Culex in 3 major genera which transmit mosquito-borne diseases. And probably the most interesting fact about it that in US, separator treated as inert ingredient and absolutely not regulated. And this is kind of more description how it works. Essentially, we're using large particle flow cytometry, and we're just using kind of, you have GFP, you basically go one way, you don't have GFP, you go another way. And with a Corpus machine, we can do sex sorting at a relatively high rate, 2 or 10 or 20 lavis per second. And then kind of this image compares us with probably the best state-of-the-art technology developed by Verily, where they use image recognition of adult mosquitoes. And they literally make mosquitoes emerging walk on a treadmill where they image with cameras. And you can imagine that processivity is quite low. They need a lot of these devices working in parallel. And here I will try to show like how fast Corpus can sex sort. And I'll see if the image video will work. So essentially what you see here, all these are tiny L1 lavis. In each of the clicking sound, it's positive selection of a male lavi. Then the female is selected, you don't even hear it, goes into a different container. But it's basically kind of very fast. You don't hear a clicking sound, but it's a tiny jobs. And then here, we added water specifically, but it's probably around 2,000 to 3,000 lavis. So it's a very efficient process. I have to go back. So the second part of technology, CRISPR gene knockout. We kind of developed this type of tool first, and we call it precision-guided stimulants technique. What essentially we did, we separated Cas9 and guide RNA into different strains. That way we inactivated CRISPR system. We can have these lines, we can homozygose them. And kind of the idea was to use guide RNA to target 2 genes. One gene is important for female viability, and if you knock it out, females are dying out. So you're selecting them out. Another gene important for male fertility, and if you knock it out, males are completely sterile. And because we don't need to have any kind of precision repair, we're just basically shredding these genes at multiple locations. In our system, we have 4 targets in each of these genes. But then we have these strains, we cross them together, we make progeny, and for Aedes aegypti, we can basically get these eggs as a progeny, desiccate them, and essentially store them. But once they develop, the system becomes active. During the development, completely hands-free, your males will be sterilized and female will die out during development. And we also speculated that because we call it precision-guided, we thought that if we're knocking out genes specifically and don't touch anything else in the genome, we will have high fitness, competitiveness, and longevity of the males. And CRISPR works almost everywhere, and now we build these CRISPR gene knockout, or PGSIT, in 4 different species, as proof of principle in Drosophila melanogastra, 2 different systems in Aedes aegypti. This is agricultural pest, Drosophila suzuki, and in Anopheles gambia. And this kind of just to illustrate some fitness data. This kind of, I was inventor of this technology, and it's very close to me. But in Drosophila, we can do very precise assessment of competitiveness. We can do single pair mating of males and females, and we can see that white bars, you have exactly the same egg number, and gray bars, it's just basically, they go down if you add another male, almost to 50%. It shows that they are very competitive. But the most strange thing was in Drosophila that the sterile males, these two lines, they live 60% longer. So not in Drosophila, it's a bizarre system. Not only we actually preserve fitness, but we actually make them live longer. Then this is kind of similar data for Aedes aegypti, and this is the same longevity. You also see that longevity is not statistically different between our kind of generated males and all type of different controls. And this image shows how effective the penetrance, because we essentially shedding these genes during the entire development. This image shows sperms, they're elongated, and there is like basically cells which are spermatids. They're not elongated, they're not moving. And in one system, which are the best so far for Aedes aegypti, we're making females flightless, and that's how we're selecting them out. And these females, they're absolutely not flying. They cannot even hold the wings properly. While male, these blue lines, they fly exactly like control, but red lines, females don't fly. And this also shows that, first of all, females of Aedes aegypti, they mate only once in their lifetime, and also that our males are competitive, because if they spend time with females like 2 hours, and then we add surplus of wild-type males, we still have some eggs not hatch. And if we spend more time, basically the bigger percentage of eggs will not hatch, and so on. And then this is kind of Anopheles gambia, which is more relevant. The progeny number, so there is absolutely no females in both directions of process, 0 of them emerge as adults, whereas plenty of males emerge. Males can mate, this mating plug, wild-type control, these PGST males mated with wild-type females. And this number of eggs, and these eggs which absolutely don't hatch from our males, but wild-type eggs hatch. Longevity also not affected. And this is also some kind of very good way to assess competitiveness. If we're adding our males, the more males we're adding, basically the fewer eggs will hatch. And so what we've done finally, now we combining separator with CRISPR gene knockout. So what it means now, Cas9 line have separator inserted in it as a construct. And it's kind of that unique separator with unique colors, and I try to use these different colors in the image. And guide RNA also have its own separator. Kind of for production, we can now sort these lines very early as L1 and put them as LAVIs. Cas9 female, male guide RNA, and once they develop, they mate only in direction we want them, and we can have a really big production of eggs. We can count eggs in the same machine, which we use for sex sorting. And we can, for deployment, we essentially just put fish food and water. And this is kind of our kind of minimalistic product container, fish food and egg, 10,000 eggs, 2,000 eggs. And this is for scale, the lid to close it, and this is kind of design of our food port with eggs. But essentially, we move along for Aedes aegypti dramatically, and with different colors of separator, we can do all type of exquisite quality control. These are not gummy worms, these are mosquito larvae, and we try to put them in the nice lines, but they're alive, they wiggle around. But it just shows what we can tell apart Cas9 guide RNA as a parental line from a progeny. And it's important because we can always hatch a small batch of eggs, confirm that both components are present, and if they're present, we know that penetrance is 100%. All the females will be flightless, all the males will be sterilized. In case of Anopheles, females will die during development. And this is kind of moving along in 5 species in the lab. We will do pilots, and I will describe them later for Aedes aegypti in the US. But for Anopheles, we're at the final stages. We already assembled plasmids, we already got some lines, and we're testing them. For Stephensi, we established lines, we have separator. For Aedes aegypti, Albopictus, we don't have separator yet, but we know the Cas9 line is working. And for Culex, we have separator. And this is kind of regulation, how in US they regulate these type of products. Separator, they do not regulate. It's considered to be inert ingredient, whereas next generation SIT, they treat it as a biopesticide, and they use the same logic, the same legal framework for chemicals. This means that for novel chemical, in our case NGSIT, in each species, we have to confirm efficacy and safety. And we can do that with experimental use permit. And we already submitted it to United States Environmental Protection Agency. And essentially we hope in this 2026 we'll run pilots in California, Florida, Texas, and Puerto Rico. But we also run pilots in 2 months, very small level, which we can do. We don't have to trigger EPA control in Texas under 10 acres, so we're going to test it very, very soon. And then this is kind of how I think basically all the technologies, genetic technologies available for controlling mosquitoes. This is conventional Wolbachia or radiation-based SIT with low suppression efficacy, very expensive, but it is completely acceptable for regulatory authorities. And then we have a GenSIT, which is super effective, very low cost, but I don't know like how you proceed with regulation, then you have to deal with multi-country level. And then we kind of in the middle, we solve scaling issue of conventional SIT. It's completely contained technology. You can apply it below these containers with eggs once in a month. It doesn't have to be done professionally. Suppression should be relatively kind of efficacious, and from all the lab studies, we basically see suppression very efficacious. And then you have to deal only with one country regulation because it doesn't spread. And sort of, as I said, we basically kind of every component is ready for next generation in Anopheles Gambia. And one thing I also start thinking that essentially all the components we have for next generation SIT is also the same components which you have in the GenSITs. So it would be very logical kind of middle step to test next generation SIT before some countries decide to proceed for GenSIT technologies. And this is kind of how I view separator. We have it working for Anopheles Gambia, for Anopheles insecticide. If any country wants to use them for radiation to optimize it or for Wolbachia control, and we actually established Wolbachia with help from our collaborators in Wolbachia company in Anopheles insecticide, now in true aggressive separator. And then second step is just more effective solution based on deployment of X or LIDs, next generation SIT, and then essentially GenSIT, where we can still help because we can use separator in a wild-type genetic ground and provide huge number of females to be able to scale GenSIT for the application, right? And we're also doing the application for GenSIT. If the separator is incorporated, you can also remove females very effectively and release only males. And this is our team. We're relatively small. There's only 6 people, and so come talk to me if you're interested. I'm very grateful for your attention and your interest. And we also got some funding from Gates Foundation. I'm very grateful to them for that. Thank you.